share the screen. I'll take a little bit of water. My throat seems to be a little off. For some reason, it's not sharing the screen. It's just taking a long time. Just a minute here. Why isn't that working? Oh, there it goes. I had to do it about 3 times. All right, today we're working on chapter 14, and we should finish the chapter today. There is no lab today. I will be logging in from 6:30 to 6:45 to answer questions. You do not need to show up unless you have questions. All right, we were talking about chapter 14, principles of disease and epidemiology. And we had just talked about the patterns of disease. Any questions about any of this? If not, let's move on, talking about disease. Some predisposing factors. There are certain factors that can make someone more susceptible to and become more affected by the occurrence of disease. For example, the shorter urethra in females make them more susceptible and getting a yeast infection at higher incidence than males. There are inherited traits such as the sickle cell anemia gene, which make you more likely to come down with disease. Another thing which isn't entirely inherited, it's partially inherited, is diabetic patients are more likely to come down with the disease. Climate and weather are more likely to affect the occurrence of disease. And I think we all know this. We all know that colds and flus are much more common in the wintertime than they are in the summertime. And I can actually state there are 2 reasons for that. The first is that in colder weather, people shut the windows and then they stay indoors more. And so there's more people together indoors. So they're more likely to spread the cold. That is true. But there's another reason why colds and flus spread more in the wintertime. And it's because they survive better in the cool weather. In the heat of the summer, the colds and the flu viruses, they die out sooner. On top of that, the summer has more intense UV from the sun, and the UV light kills the DNA or RNA of the viruses. And so that means that the viruses are more likely to die in the summer from the UV light as opposed to the winter. Fatigue can also affect your susceptibility to coming down with disease. And since you guys are students, you guys are probably often fatigued. So you should watch that and try and get as much sleep as you can to help your body resist infection and fight off infection if you get it. Age is also a factor in getting disease. I think everybody knows with COVID-19, you're more likely to become sick and then more likely to become seriously sick or even die with COVID-19. But in most diseases, it's the young and the elderly who becomes sick or more seriously sick from disease. With COVID-19, it's just the elderly. You have to get very, very young, like newborns, before COVID-19 becomes more of a serious factor. But in most diseases, the young are more susceptible to getting diseases, and it's more serious than other age groups other than the elderly. Obviously, the elderly and the very young are more susceptible and disease is more serious. Lifestyle can also affect whether you come down with the disease or not. And I think we know some of these. That is, people who have multiple sex partners are more likely to come down with the disease than people who are monogamous or what do you call it, zero, whatever that number is. And then people who smoke are more likely to get a respiratory disease. And it's because the smoking damages the mucus lining in the respiratory tract. And so the point is lifestyle can affect whether you get a disease or not. And then chemotherapy and other harsh hospital treatments can affect whether you're more susceptible to getting a disease or not. Chemotherapy tends to weaken the immune system. And if your immune system is weaker, you're more likely to come down with the disease. Any question about any of that? Surprisingly, the development of a disease tends to follow the same course regardless of what disease it is. Once the host defenses are breached, disease development follows a certain sequence. And this is similar for both acute and chronic diseases, serious diseases and mild diseases as well. All diseases have an incubation period. The incubation period is the time from where you first get the disease-causing organisms and the time you first have signs or symptoms. That's the incubation period. The time where the organism is growing in you and gets to a number. If you look at this chart, it's the number of microbes on the y-axis and also the seriousness or the seriousness of the signs or symptoms on the y-axis and just the time on the x-axis. The incubation period is followed by the prodromal period. Like the prodromal period is when you have mild signs and symptoms. An example would be when you're first coming down with a cold and you say, geez, do I have a sore throat? And you have to think about it before you say, yeah, I've got a little bit of a sore throat. That's the prodromal period. The period of the illness is when the signs and symptoms are at their most intense. It's no longer mild. And so instead of thinking about it, do I have a sore throat? You say, geez, now I have a real sore throat. The period of illness is followed by the period of decline. That is when the patient starts getting better. If you have a fever, the period of decline is when the fever breaks or the fever ends. On something like a cold and the flu, it would be probably when your nasal drippage starts thickening and becoming less frequent, when you start feeling a little better. The period of decline is followed by the period of convalescence. The period of convalescence is when you still have the disease-causing organisms, but their numbers have fallen below a threshold number, and you no longer have signs or symptoms of disease. So you still have the disease-causing organisms, they're just at a low number below the threshold, so they're no longer giving you signs or symptoms. Talking about the period of convalescence, I noticed that sometimes when I get a cold, it doesn't work with the flu, I've tried. When I'm first coming down in the prodromal period, if I get lots of rest, and I'm talking about excessive amounts of sleep and rest, I can knock the illness from the prodromal period to the period of convalescence, and I'll talk a little bit more about that in a minute. What I'm doing is that when you're sleeping, your immune system is at its strongest, and you're boosting your immune system when you're sleeping. And when the immune system is stronger, it can knock down the disease. And what I do is I knock the disease down below the threshold number where I no longer have signs or symptoms of disease. And frequently when I'm feeling better, I stop getting all that rest, and then I start becoming fatigued, and then the disease comes back on me. And I said, oops, I've still got it. I got to get more sleep. Now, this is something that I can do because I'm an instructor. I realize some students wouldn't have the luxury of sleeping a lot. But if you can do that, you can knock the cold from the prodromal period, making it skip the period of illness and the period of decline and move into the period of convalescence. It only works with the cold. It doesn't work with the flu. And then that brings up the topic that I needed to talk about, and that is diseases do not have to follow all of these 5 stages of disease exactly in each stage. As I stated with the cold, you can move it from the prodromal period to the convalescence period. Okay. And then there are some illnesses where you have an incubation period and you move directly into the period of illness. Sometimes when I've had the flu, I'm fine. I have no signs or symptoms. And then the next moment, I don't have any mild symptoms. It's like somebody hit me over the head with a sledgehammer. I have very severe signs and symptoms immediately. So some cases of the flu skip the prodromal period. And then there are some illnesses which skip the period of decline or the period of convalescence. Can anyone think of a disease where that happens? Not hearing anything. Well, what about a disease where the patient dies? They never leave the period of decline, illness. And it used to be that AIDS patients entered the period of illness and then they died. Nowadays, they can take medication and live for a long period. And they can actually leave the period of illness and go into a period of decline. There are at least some patients who even enter the period of convalescence. So the point is that you don't have to go through all of these stages of disease. You always have the incubation period, I guess. And that one's always true. And I suppose for every disease, you always have a period of convalescence. Okay. But all the others may be there or they may not be there. Generally, an illness will go through all 5 stages, but they don't have to. Okay, here's a question for you. In which period of disease are you no longer communicable? Is it the period of incubation, the prodromal period, the period of illness, the period of decline, or the period of convalescence? What's your guess?
The period of decline.
That's a good guess, but no, that's what my mother always said. I'm getting better now. I'm no longer communicable. And I'd say, no, mother, you're talking to a microbiologist. I know better. You're in the period of decline or the period of convalescence. And as you see from this graph, you have the microbes in all of these periods. So you are communicable in all of the periods. For some common diseases like the colds and the flus, or at least for adults, you are most communicable or contagious for these 2 diseases late in the incubation period and in the prodromal period. You don't need to know why an adult is that way, but in the period of illness, the adult's immune system is ramped up and is putting a a brake on the disease-causing organism. And so for the colds and the flus, adults are actually less communicable in the period of illness than in the prodromal period and late in the incubation period. That doesn't mean they can't spread it. I just said that they're less likely to spread it. It doesn't happen that way with children, and my guess is that adults have this system to try and prevent giving the disease to their children. Okay, so it's probably an evolutionary factor, but you don't need to know that. I'm just telling you that with many diseases, common diseases, it's actually late in the incubation period and in the prodromal period when we are most communicable. But we are communicable in every period. Okay, so that's something you should know. Even when you have no longer signs or symptoms of the disease, you are communicable. All right, let's talk about the spread of an infection. In order for an infection to spread, it has to have a reservoir. A reservoir is a source of infection. Reservoirs are continual sources where the disease organism can be found. The reservoirs can be living or non-living. Human reservoirs are a major reservoir for human diseases. And obviously another human can have a disease-causing organism and then will be a reservoir for spreading that disease. There are some diseases which humans are the only reservoir. For example, AIDS and gonorrhea, humans are the only reservoirs that other humans get the disease from. Now, it's theoretically possible that a human could get HIV from a chimpanzee, but that's very unlikely, and we have no cases known for that. Although you could always argue that the first human case got HIV from probably a chimp, and so it's possible. But generally speaking, for these two diseases, humans are the only reservoirs. There are animal reservoirs for certain human diseases, and some diseases like rabies, for example, and Lyme's disease, humans only get these diseases from animals. A rabies from a biting animal, a Lyme disease from a biting or blood-sucking insect or arthropod. Zoonoses are animal diseases that can be transmitted to humans. And rabies is a classic example of a zoonoses, an animal disease that can be transmitted to humans. If you don't know, rabies, once it gets into a human, it's a dead end. Rabies does not spread from a human to other humans or animals. Meaning that if a rabid human were to bite another human, he would not transmit the rabies. Lyme disease is not that way. A tick can bite an infected human and then get infected and then bite another human. So Lyme disease can spread, but rabies doesn't spread once it is in the human. So it's a classic case of a zoonoses. There are non-living reservoirs, and various diseases like botulism and tetanus come from non-living reservoirs. Non-living reservoirs can be the soil, water, and then lettuce. And they're counting lettuce as non-living because once you pick it, it's gonna die. Any questions about any of that? The transmission of disease can happen by three principal routes by which the causative agent of disease is transmitted from the reservoir to a susceptible host. There's contact transmission, such as kissing or handshake. Vehicle transmission, that is transmission by an object. And then there's vector transmission, transmission by an animal. Contact transmission has three types. There's direct contact transmission, and that would be a case of person-to-person transmission, requiring close association between the infected individual and a susceptible host. For example, somebody sneezes on their hand, and then they shake hands with someone. Kissing is another case of direct person-to-person transmission. Indirect transmission is the spread from the reservoir to a susceptible host via a non-living object. We call a non-living object a fomite. An example of this could be someone sneezing on their hand, giving the disease-causing organisms on their hand, and then they touch money. And then somebody else touches, they pay for something with the money, and then somebody else touches the money, and then they get the disease-causing organism. Another case, which I'll show you in a minute, is a baseball can act as a fomite, causing the spread of indirect transmission. The third contact transmission is droplet transmission. This is transmission via an airborne droplet that travels less than 1 meter. It's different than airborne transmission. It's called droplet transmission. And I'm not going to test you on this because I think droplet transmission and airborne transmission is an arbitrary distinction. I'll show you in the picture here. And like in a sneeze, if you're within 1 meter, this region here, that's droplet transmission. If you're at 1 meter and less than 1 inch, it's airborne transmission. And it's the same sneeze. So like I said, I'm not going to test you on that because that's, to me, an arbitrary distinction. Okay, but contact transmission can be direct. It can be indirect, like the baseball. Somebody sneezes on the baseball or sneezes on their hand, then they touch the baseball, then they throw the baseball to the catcher, and then the catcher picks it up on his hand. Now, actually, if you were to sneeze on your hand and touch the baseball, and the catcher were to touch the baseball with his hand, that wouldn't give the catcher the disease-causing organisms. He has to then inoculate one of his mucous membranes on the face, the eyes, the nose, and the mouth. Occasionally, he could inoculate the ear as well. But the point is, it has to go from the hands to a mucous membrane on the face. But unfortunately, humans are very good at transmitting diseases, especially colds and flus, indirectly. If you don't know, most colds and flus are picked up by your hands. Like when somebody touches a doorknob and they pick up the virus, and then we inoculate ourselves by touching one of the mucous membranes on our face. So indirect transmission is a primary way that colds and flus spread. Obviously, droplet transmission is another way, and airborne transmission is a third way. And like I said, I'm not going to draw a distinction between droplet and airborne. If you don't know, COVID-19 can easily spread within 6 feet and 1 meter. Is that what I said? Yeah, 1 meter is only about 3 feet. So COVID-19 spread 6 feet. So it's both droplet transmission and airborne transmission. we'll talk more about COVID-19 a little bit later in the term. Any questions about the 3 contact transmissions? All right. Vehicle transmission is when an inanimate reservoir spreads the disease-causing organism from the source to a susceptible host. And the inanimate reservoir can be food, water, body, fluids. It can also be, no, that's indirect, an object. Okay, so waterborne transmission is when water spreads the disease-causing organism, such as contamination of water by sewage. That doesn't happen very much in the first world, but in the third world, this is a real health problem. A foodborne transmission, like if somebody sneezes on your food and then gives you the food, or even if they sneeze on their hands and then they, I don't know, pick up the fries and put them in your basket, foodborne transmission. And then airborne transmission, this is when you sneeze and the droplets travel more than 1 meter. You can see why I don't really want to talk about airborne transmission and droplet transmission, because the same sneeze is both a vehicle transmission and a direct transmission, the droplet. And so I'm not going to test you on that. All right, any questions about vehicle transmission? A vector transmission is when an animal carries the disease-causing organism from the source to the susceptible individual. The most important group of vectors are the arthropods, especially fleas, ticks, and mosquitoes. The transmission of disease can be a mechanical transmission or a biological transmission. In mechanical transmission, it's a passive transport, such as this fly picks up the disease-causing organisms, the microbes, on its feet, and then the fly deposits the disease-causing organisms on a piece of food, for example, and then the food is consumed by a human. So the human gets the disease-causing organisms by consumption of the food, by mechanical transmission. Any question about that? Biological transmission is where the pathogen reproduces inside the vector. And a good example of biological transmission is malaria being spread by a mosquito. The malaria is picked up by the mosquito, and the malaria reproduces inside the mosquito. Then when the mosquito bites a human, the malaria comes out of the mosquito and goes into the human. So that's a case of biological transmission, mosquitoes and malaria. Any question about any of that? If not, we're going to change topic again and talk about nosocomial infections. These are also called hospital-acquired infections. A nosocomial or hospital-acquired infection is an infection acquired by a patient as a result of a hospital stay, meaning the patient did not have the disease-causing organisms before they came into the hospital-like setting. And then they got the disease-causing organism in the hospital-like setting. When I'm saying hospital-like, it could be a hospital, or it could be a clinic, or a nursing home, something like that. It's estimated that 5 to 15% of all hospital patients in the United States come down with a nosocomial infection. This is 2 million people per year in the United States, and it causes an estimated 20,000 deaths each year in the United States. So it's actually the 8th leading cause of death in the United States. And I assume that's close to about the case even with COVID-19, because I think COVID-19 is above the 8th leading cause now. So over 1 in 20 hospital patients comes down with the nosocomial infection. It used to be about 10% of the patients, but recently, because hospitals have been really trying to reduce the number of nosocomial infections, they have reduced the number down to about 5%. And that was the latest number I'd read about. About 50% of nosocomial infections are preventable. And you don't need to know much about this, but it's a major source of infection, a major cost in the American healthcare industry. Nosocomial infections are a result of the interaction of 3 factors. 1, microorganisms are present in the hospital or hospital-like environment. That's unavoidable because obviously everywhere we go on Earth, we're going to have microorganisms. But hospitals are a major reservoir of sick people, and sick people have organisms which cause disease. So some of the sick people have, communicable diseases, which can be a nosocomial acquired infection. There's also some normal microbiota, which are opportunistic pathogens. And some nosocomial pathogens are antibiotic resistant, in which case a nosocomial infection would become very severe because the antibiotic resistant nosocomial infection will be treated with the standard antibiotic and it won't work. And then the doctor will switch to a second antibiotic and it also doesn't work because the nosocomial infection is resistant to antibiotics. And the doctor said, oh, the second one didn't work. I guess I'll have to screen the patient and see what works because they seem to be resistant. Well, by this time, the patient may die because you're in the hospital for a reason. You're not likely to be a good, healthy patient if you're in the hospital. Any questions about that? And in a sense, this is another reason why nosocomial infections spread in the hospital. The patient is in the hospital for a reason. So they are usually a compromised host, one whose resistance to infection is impaired in some way, impaired by disease, by the therapy the patient is receiving, or by burns, or by trauma. Broken skin or broken mucous membranes compromises the host. The patient will be more likely to come down with an infection. Burns, injections, ventilators, urinary catheters, even the administration of anesthesia makes the host more susceptible to coming down with a nosocomial infection. And then some patients are immunosuppressed. Various drugs, radiation therapy, and burns weaken the host immune system, making the patient more likely to come down with a nosocomial infection. Third, in the hospital, there's a chain of transmission from the source to the susceptible patient. There are 3 principal routes of transmission. There's direct contact transmission, and that can be patient to patient, such as a patient in a special care unit. It can be hospital staff to patient, meaning hospital staff touches an infected patient, and then they don't wash their hands and they touch another patient. They can spread disease that way. There's indirect contact transmission that happens via fomite, urinary and IV catheters, contaminated surgical dressing can be indirect contact transmission. This used to be a major way of getting a nosocomial infection. If you look at the older movies, and the doctors in the 60s, they proudly wore their stethoscope on their chest, and then they would come up to a patient and listen to their heart, putting the stethoscope directly on their skin. And then what would they do when they're done? They'd drop it and it'd thump against their chest, and then they'd move on to the next patient and then take the stethoscope and listen to the next patient's heart. That was a good way to spread nosocomial infective agents. And in the past, Staphylococcus aureus was actually the leading nosocomial infective agent because Staphylococcus aureus was on the skin of 20% of Americans. And so it'd be picked up from the hospital staff or the doctor, like on the stethoscope, and then spread patient to patient. There's also vehicle airborne transmission where the ventilation system can spread the transmission. Hospitals are aware of this, and especially with airborne diseases, they try to put a patient in a room where the air can be sort of filtered, like with COVID-19. An even better example was, what was that one? Ebola. There are various ways to control nosocomial infections, and I should state that this is one of the most important topics that you will learn in microbiology, because most of you are going to go work in a healthcare facility. That's why you're taking this class. It's what you want to do. And so you have to learn to control nosocomial infections. And so if you learn that, you'll reduce the spread of nosocomial infections. And it'll be a major thing that you can learn from this class. And I actually had a person tell me what she did was she did the lab tests on the people who came down with infections. And she said that nosocomial infections were the problem of stupid people spreading around nosocomial infections. And we can control at least 50% of them by just taking general precautions, like always washing your hands after you touch a patient or replacing the gloves. Always doing certain things. In microbiology, you learned aseptic technique. When you go into the hospital setting, you should always apply aseptic technique to stop the spread of nosocomial infections. You should have proper handling of contaminated materials so that the microbes stay inside the tube or the culture and don't spread beyond the tube or the culture. Something that's been shown to reduce nosocomial infections by 50% is simply frequent and thorough hand washing. If you educate the staff, the patients, and the visitors, we can reduce nosocomial infections. And hospitals have gotten very good at this. There will be a sign, usually at the front door or in the incubators, saying, please sanitize your hands before you go and visit a patient. And then there's sanitation stations almost at every patient's room, in the elevators, and usually when you first come into the hospital. So this has gotten much better. I remember in the old days where there was no education for a visitor or for patients, and there was no way to even sanitize your hands other than going into the bathroom and washing your hands. There are some other procedures you can use to controlling nosocomial infections. I'm going to refer you to seeing the online chapter notes. And that's what do you call that? Not the objective, the notes for this chapter. Go take a look at those, and it lists a few other things you can do for controlling nosocomial infections. All right, any question about any of that? How are we doing time-wise? We're doing well. If there's no questions, let's change topic again and switch to epidemiology. Epidemiology is the study of where and when disease occurs and how the diseases are transmitted in populations. The CDC collects and analyzes US epidemiological information. And then they publish that weekly in the Morbidity and Mortality Weekly Report. The MMWR is what that's abbreviated, because Morbidity and Mortality Weekly Report is a difficult thing to say. And you can find that at www.cdc.gov. And here I'm going there to try and show you where it is. If you see it, go ahead and point it out to me. Oh, there's a note about COVID-19 pandemic. I'm going to have to get another link there because I'm not seeing the link anymore on their front page, which is really weird. Oh, there it is, right there. MMWR. Current week. That's not really what I wanted to see. I wanted to see. Oh, there it is. No, that's not it. Well, here's one thing. But anyways, they publish that each week, giving the morbidity in the United States and the mortality. Morbidity is the number of people who have become infected. Mortality, the number of people who die each week in the United States. Notifiable diseases are the ones that they're collecting their data on and then publishing. Notifiable diseases are those diseases that physicians are required by law to report. The CDC is a branch of the government. Usually the physicians report to a local county health organization, the local county health. And then the local county health reports to the state health facilities. And then the state forwards it to the Center for Disease Control. Morbidity is the incidence of a specific disease, and we're talking about notifiable disease. The morbidity rate, the number of people affected in relation to the total population in a given time period. So the morbidity of COVID-19 is frequently published. And what the heck is it now? I'm trying to remember, is it 700 million who have been infected or 700 million who have died from COVID-19. And I'm sorry, I don't remember that number now. I used to know this. I think it's 700 million people have died from COVID-19 in the United States alone. Mortality is the number of deaths from a notifiable disease. The mortality rate, the number of deaths from a disease in relation to the population in a given time. So usually only the notifiable diseases are reported to the Center for Disease Control. All right, any question about any of that? Any questions at all about chapter 14? I guess I should save that. Excuse me. If not, let's move on to chapter 13. Viruses. We'll just get started on it. Talk about a few slides and then we'll end early today. So chapter 13 is discussing viruses, viroids, and prions. As usual, I give you the first slide, which gives you the major goals of this chapter and a rough outline. Know the terms. What's a capsid, an envelope, a spike? What's a bacteriophage? What is a lytic life cycle, a lysogenic life cycle? What's a prophage, a provirus? What's a latent life cycle, a persistent infection life cycle? What's a prion and what's a viroid? Understand the characteristics of viruses, their structure, and their classification. Understand viral multiplication. And then lastly, understand about prions and viroids. Any question about any of that? The only goal of a virus is to replicate. How do viruses do this? Their general strategy is to invade a host cell, hijack the host cell, and take it over. Force it to make new viruses, which then escape from the host cell. Why don't I, oh, I'm on the wrong one.
In the microscopic world of bacteria, a miniature act of piracy is about to take place. T4 bacteriophages. With their spider-like tail fibers and prism-shaped heads, these viral marauders plan to hijack an unsuspecting bacterium and use it as their own metabolic nursery. This E. coli bacterium has seen its last day. Binding to its surface, the virus injects its precious cargo of genetic material into the cell, commanding it to produce a new batch of T4 bacteriophages. In a final act, the bacterium is forced to self-destruct, releasing hundreds of viruses ready to kill again.
Okay, any questions? Some general characteristics of viruses. When viruses are not within a living host cell, they're generally inert. They're not metabolically active unless they are in a host cell. So viruses display little or no metabolic activity outside of a host cell. They require a living host cell in order to multiply. So we can say viruses are obligate intracellular parasites. They only multiply within a living cell using the host cell's enzymes, nucleic acid, amino acids, and other molecules to replicate. A virus has no ATP generating mechanism. They get all the ATP they need from their host cell. Viruses do have a genome, but that genome can contain either DNA or RNA. Never does the virus contain both DNA and RNA. It either has one or the other. Viruses have as their central molecule their genome, and the central molecule will be surrounded by a protein coat. So all viruses have their central genome, which can be either DNA or RNA, and a protein coat that we call a capsid. Some viruses, not all, but some viruses have an outer lipid envelope surrounding the protein coat. There are some general characteristics of viruses, and that is they have a host range, the range of host cells that they can infect. And we're going to talk about different species being the range. The host range is determined by specific attachment sites on the host cell, as well as on the virus. So you have specific receptors on the host cell and specific ligands on the virus, or attachment molecules on the virus, which are ligands. And they have to link up, meaning be, what's the word I'm looking for? Corollaries before they can bind. Most viruses only infect the cells of one or a few species. Like COVID-19, that's a bad example because it can infect cats especially. HIV would be a better example, and that is it generally only at least human HIV, only infects human cells, and actually only certain cells of humans, like the brain and helper T cells. It doesn't tend to infect skin cells, for example, cells of your nose. So most viruses only infect the cells of one or a few species. HIV can infect one chimp. I don't know which one there is. There's two chimps. Pan troglodytes is one. The second one, I don't remember, pan something or other. It can infect one chimp, but it doesn't cause AIDS in the chimp, meaning it doesn't impair the immune system of the chimp. So the virus can infect the chimp, and I assume the virus grows in the chimp, but it doesn't give the chimp AIDS like it does in human. Anyways, the specific binding of the viral factors to the host receptor factors makes the virus specific and determines the host range of the virus. Most viruses, like I said, infect only one or a few species, but there are some which infect many. Some of the encephalitis viruses, like Western and Hawaiian encephalitis, and Some of the flu viruses can infect humans, horses, pigs. Every once in a while, they can infect cats and dogs, depending on the flu. And then they can also infect many birds. So there are some which don't follow that pattern, but most viruses do. Bacteria viruses are given the name bacteriophages, or sometimes just phages or phages. I'm not going to use the term phages or phages. I'll use the term bacteriophage or bacterioviruses. Any question about any of that? Viruses that infect a bacteria only infect bacteria cells, and generally they only infect one or a few species of bacteria. So bacteriophages cannot infect humans, and we'll talk a little bit about that later. When we're talking about viruses, we should talk about the size of viruses. But I think this is a good time to stop. So I'm going to stop right here and go back and say, are there any questions? All right, if there's no questions, I'll see you next Tuesday, unless you're coming to the question and answer period at 6:30. Come on.